Howdy, this is Lab Report 7, Biochemical Test 1. This is Group 2, Diego Castillo, and myself, Rita Cantu Mahurin. In our introduction, we focused on the purpose of this lab, which was the 4 key biochemical tests that we use to identify and differentiate gram-negative bacteria based on their metabolic capabilities. We used the indole methyl red, or MR, the Voges-Prosker VP, and then the citrate test to assess the tryptophan breakdown fermentation pathways, acetone production, and citrate utilization, respectively. These tests were performed on E. coli, E. cloacae, and a gram-negative unknown organism to help determine metabolic patterns and contribute toward identification of the unknown bacterium. The media we used were the tryptophan broth for the indole test, the MRVP broth for the MRMVP test, the Simmons citrate agar slants for the citrate test, and then the bacterial cultures included the E. coli, the E. cloacae, and then our unknown gram-negative organism. The supplies and equipment were the inoculating loop and inoculating needle, the Bunsen burner or sterile work area, and then the incubator. The pipettes are droppers, and then the reagents we used were the Kovacs reagent, the indole test, and then the methyl red indicator, the VP1 and VP2 reagents, and then our test tube and test racks, as well as our PPE, which are our gloves, lab coat, and goggles.
Here are the results for the indole test. Our E. coli, you can see it turned a pink color, meaning it is a positive. The E. cloacae did not change. It stayed a kind of clear-ish color on the top. And then for our unknown 292, it was the same as the E. cloacae. The E. coli has that red ring at the top because it means it is a positive, meaning it produces indole from tryptophan. The no color change means the E. cloacae is a negative, and from what is seen from our results, the unknown 292 is also a negative. The principle behind the indole test is to detect the ability of the bacteria to split tryptophan into indole, um, into indole pyruvate and ammonia using the enzyme tryptophanase. The reagent used is Kovacs reagent. A positive result shows that red ring at the top of the broth, like seen in the E. coli. Um, the inference E. coli can produce indole, E. cloacae cannot, and the unknown also means it cannot if the results are correct. Here is the methyl red test. The E. coli is a red color after the reagent, meaning is that it's a positive, meaning there is mixed acid fermentation. The E. cloacae should have been a yellow or orange. It is a lighter color, you can see kind of from the E. coli, but it is still a little difficult to see, meaning it should have been a negative because it does not use the mixed acid pathway. And for unknown gram-negative, it looks more like our E. cloacae than our E. coli. So the metabolic pathway that's being detected is the mixed acid fermentation pathway. The reagent uses methyl red, and a positive result is which is a red color, indicates stable acid production from glucose metabolism. E. coli uses that mixed acid fermentation. E. cloacae uses butan, butanediol fermentation, and the unknown fermentation type depends on what we see. It is a little difficult to see, but it could look like the E. cloacae, meaning it uses that same butanediol fermentation. Here's the Voges Proskure test, or the VP test. Our E. coli has no color change. Our E. cloacae has no color change, and our unknown 292 has no color change. The E. coli is for sure a negative, meaning there shouldn't have been a change in color. That means it does not produce acetone. However, for the E. cloacae, it should have been a red color after the reagents. It is a little bubbly. That is from us trying to fix it and see if we could get results. Maybe the bacteria wasn't touching the right spots, but it should have been a positive producing that acetone, a butanediol pathway. The gram-negative has no apparent color, so it could mean it's a negative or the test wasn't correctly done. The butanediol fermentation pathway is the acetone production. The reagents used were VP1 or alpha naphthol. and VP2, KOH. Um, it complements MR, the methyl red, because it detects that strong acid production, while VP detects the neutral end products like the acetone, and typically organisms are positive for one and negative for the other, making the two tests complementary. So as we said, the E. coli for sure is a negative. That's why the methyl red was a positive, and the E. coli should have been a positive, but it didn't appear so. These are the results for the citrate test. Our E. coli stayed a green color. The E. coli and unknown 292 came out blue, meaning those are positive. Our E. coli is negative. What this means is that the blue color changes are from alkalization of using that citrate that is in the results. And since it is blue, that means it can use the citrate as its sole carbon source. And if it's green, that means it cannot use the citrate. So E. coli can't use citrate as the sole carbon source, while E. coli and our unknown 292 can. Um, and like I said, the blue color indicates that alkalinization due to citrate utilization. So the E. coli can't use citrate and the E. coli can.
In conclusion, by performing the indole MRVP and citrate tests, the metabolic characteristics of E. coli, and the unknown gram-negative organisms were observed and compared. Each biochemical test revealed specific enzymatic pathways, such as the indole formation, the type of glucose fermentation, the acetonin production, and the citrate utilization, and collectively, they aid in the identification of the unknown bacterium. These biochemical reactions form the foundation of bacterial differentiation in microbiology, demonstrating how metabolic profiles can be used to classify organisms and support clinical and research-based microbial identification. Thank you so much. Once again, this is Group 2.